When I first started talking about multiple sclerosis and genetic factors in multiple sclerosis, people thought I was a bit nuts, and it was a very hard battle because at that time nobody thought that there was any genetic component to MS, but people did know that if you looked at families, you could have more than one person affected. The database started with a grant from the Multiple Sclerosis Society of Canada in 1993, and it allowed us to collect DNA samples and family history information and clinical information and demographic information from all the established MS clinics across Canada.
Many genes have been identified to have a role in multiple sclerosis, but their contribution to disease is rather small. What we're trying to identify is one mutation of major effect that causes disease within families. We pull the DNA samples from the freezer and we sequence the entire coding genome from these patients to identify all the mutations that they had in their genes. Once we have found those mutations, we try to identify those that were exclusively in the patients and not in the healthy family members and not in the general population to find the mutations that actually cause the disease in these families. This mutation was identified in 2 families from Canada, and in these 2 families, 70% of the people who presented the mutation developed multiple sclerosis. This is the first time that we identified a mutation that actually causes the disease.
When Carl first came to me with these findings, I was very skeptical because I've been in this field so long. And although we've had many families where MS seems to go from generation to generation, we've never been able to find a gene change that is specific to the cause of MS. And I have to say that I was sort of vindicated because I've been saying all these years that there may be some families where genetics are more important than in other families, and I've passed many decades with people almost laughing at me. I was actually a little proud of myself that I stuck to my guns all these years.
With the genetic mutation that we have identified, we can now create animal models which are very likely to mirror the biological processes that cause disease in people. These models will be an outstanding tool for the development of new treatments which are likely to tackle the causes of disease rather than try to manage the clinical symptoms.
We could even perhaps predict who may go on to develop MS and perhaps be able to do some treatment before MS hits the clinical stages.